Here is how to look for your gene's expression using the publicly available single cell sequencing data. There are already a few websites, but this one is the latest. I think it came like a week ago. Now it's 2025 November. It came like a month or a month ago, maybe. So it's from this article, if you want to read the article. So this is the website. What's the aim of using this website? If you know the ID of your gene, you put it here, and then this is showing the transcriptome response in Arabidopsis developing seeds. So this green thing will become an embryo as it matures. So this is 7 days after pollination, 5 days after pollination, 3 days after pollination. So this will become the seed, and the seed is consisted of many different layers. Each layer have a name, and then it's denoted by a different color. When we put the gene we are interested in, it shows us which tissues it's expressed, like in the inner tissue, in the embryo, or during development, how this expression changes. This is, I think, very important information because, for example, if your gene is responsible for synthesizing one chemical, then it will tell you in which cell layers this chemical is expressed. Then what? Probably this expression should overlap with the transporter of this chemical, for example. If you're looking for a transporter of this chemical, you can examine where the gene responsible for the synthesis of chemical is expressed, and then you expect your transporter to overlap this expression, and then you may decrease the list of transporters, and then you can obtain the knockout lines of these transporters and see if the concentration of the chemical changes, for example. Okay, so what is the approach here? How did they prepare such a graph? And then I will tell you what each thing means. To prepare this graph, what they did was they took Arabidopsis plants. They had 3 replications, and they took samples from 3 days after pollination. How they do it, I'm not exactly sure. There should be like 2 ways, but yeah, I'm pretty sure they didn't do laser dissection. Sometimes they cut these cells with a laser, but I think here they didn't do that. What they did was they just destroy the cell walls, or even they didn't do that. They just take all the nucleus from every cell in 3 days after pollination, 5 days, and 7 days after pollination. So they had lot of nucleus, and then they extract RNA from these nucleus, and they already know marker genes for these layers, for every layer at every stage. What is a marker gene? This is a gene that's already characterized in the literature, and you can search and find an article related to it. Probably there was a gene GFP fusion, and they show that the gene is expressed exactly in that layer and exactly at that stage of development. So they used these marker genes in order to classify which cell belongs to which tissue and which stage. Sorry, which stage is already known, but which tissue? Because they took all the cells of this, but they don't know which tissue the cells belong. They used the reference genes, marker genes, to associate with the tissue. I don't know if it sounds clear or not. Yeah. Yeah, they did RNA sequencing for nucleus for each cell. And then they had a lot of data, and there are many different ways they employ here to show us this data. And this is something I like with this website. The other website, sometimes they don't have this a lot of data. So first, you can search 3-day old genes, five or seven. You should decide it. Let's say we search 3 days old. Okay, and then we here write the gene of our interest, but I will first show you what each of these things mean. So now we have the expression data of 3 days old cells, three DAP cells. Here, every spot corresponds to a whole RNA sequencing result. So every spot is a whole RNA sequencing result of just one cell. So if we have like 4,000 dots here, it means they did 4,000 RNA sequencing. And why some dots are further from some others? That's because the gene expression patterns are different. So if gene expression patterns are closer, then they tend to group together. So here, for example, there is a group. Here there's a grouping. 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 And you can immediately say the grouping here is quite different than the gene expression of this is much more different than the rest compared to, for example, this one, because this here gene expression is not extremely specific because there are many other groups around, or this one. But for example, this one is really far away from the others. So you can deduce things like that. So here we can select three levels of resolution. Uh, for example, the first level shows us only embryo, endosperm, seed coat, funiculus, and ovule. So seed coat is all these layers, but they don't care in this mode, this level one. We see all the expressions in the seed coat. So all these RNA-seq data belongs to seed coat. So there are so many cells that we have RNA sequencing for the seed coat cells, but much less for the funiculus, which makes sense because funiculus is this part. It's very small. And embryo is this part. It's also very small. So no wonder why we have very little amount of seeds compared to seed coat ones that are sequenced. From the same data, if we now do seed say level 2, what it does is this bulk seed code now is differentiated into OE2, OE1, EE1 because they already know how some genes should be expressed in these layers, and they analyzed those cell RNA sequencing, and they uh, the cells that show correct expression, the expected expression level, they are put into these clusters. And we immediately see the groupings in the seed code already associated with OE2, OE1, EE1, and they are different from each other, which should also already be expected. I mean, it's not the case like some of the EE1 cells are here, then it would be unexpected. We can even go further details. So even in the same cell layer, for example, um, now I don't remember that well, but I think this was OE1. Sorry, for example, 25 onwards is OE2. So this region belongs to this OE2, right? So normally what a tissue is like group of cells doing same thing, but somehow there are still subclustering in the tissues, and this information can be almost only gained with this snRNA technique when we have subclustering with some really marker genes. Like, for example, this one is known to be expressed in OE2, this one is known to be expressed in OE2, and this one is known to be expressed in OE2, but we already know they have slightly different expression uh, localizations and patterns. And using these genes as now marker genes, we can have level 3 level of resolution. If in the future you find a gene which has a very different expression pattern in even, OE2 S phase genes, for example, even in S phase, it has a very different pattern. Then you can analyze the whole data again, and then you may even build a new subcluster. And then you can find some genes whose expression is more similar to the one you find compared to the one which is shown here. I hope it makes sense. So this is what is most important. You examine your gene of interest, where it is expressed, in which stage it's expressed, and which gene is it more similar to, even, And then what information does it give you? Maybe if so there may be some transcription factors, for example. For genes expressed here, there should be a transcription factor, maybe different than genes expression here, maybe different than genes expression here, but maybe they share the same transcription factor and so on. So you can make deductions like this. Here is another feature which is, I think, less important. For example, which stage, so these are actively dividing cells, all of them. Which stage are they in, Like, are they in S stage, M2 stage, G stage, and so on? So if you do this, then it examine all the metadata and then give you this. So most of the cells, when the experiment is done, was in G1 phase. I think it makes also sense. I'm not expert in it, but probably G1 phase is the longest. And there are a lot of dividing cells. And probably you can even say which ones were embryo. Maybe embryo cells, like they are at this day, they are dividing more rapidly, for example, compared to fifth day and so on. You can make deductions like this. How do they know which stage the cells are in? Because there are marker genes, marker expressions for genes. For example, if it is S phase, let's say gene X is expressed a lot and then it decreases. And then they examine this, like at day 3, this gene is expressed in this cells, and in day 5 it decreases in this the same cells. Then these cells should be in that stage, this kind of thing. Okay, there are other things you can do. Like, for example, all the cells here now, 3 days after pollination. But if you push here, then it gives the result of all days together. So here you can see day 3, 5, 7 together. You can have an idea like how many cells are sequenced for each, and then you can say, for example, I guess 3 days you have the least number of cells. It looks to me so, but anyway. And you can see they are grouping, the expressions, the similarity of expressions. They are grouping in 7 days is more similar to 5 days. The 7 days of OE1 is more similar to, if this is OE1 again, probably yes, because it has the same shape. 7 days of OE1 is more similar to 5 days of OE1 and so on. I think this is how it should be interpreted. Then they have another thing they did, but not every website does it. They have a differential expression table. I'm again not expert how to use it, but most probably if you select um, Atlas or like day three, and then let's say you are interested in genes expressed in OE1, and then you want to compare their expression with the EE1, and then you choose here EE1, and then it filters for you which gene's expression is most different in OE1 compared to EE1. For example, this gene's expression is much more different. This is the highest. So this is the gene that's expressed most distinctly at 3 DAP. Yes, sorry, I should put here OE1, I think, also. Yes, I think now this is like how they are different from OE1. No, I didn't put the gene. No, we don't put any gene here. Alternative genes. Okay, I should admit I don't know how to use it, but somehow it shows transcriptional gene differences, the difference in transcription. So it shows the differentially expressed genes, but I've never used this for that one. Okay, and this is how to show the data once you enter your genes. Sorry, now I notice how it works. So for example, this is now EE1. Now if I put my gene of interest, let's say I want to examine FLS1. I know this already, this gene. So I write here FLS1 Arabidopsis tear to obtain the gene number. And here it's a flavonol synthase gene. I take this number, put it here. They don't have copy paste, maybe they have. Okay, I chose here. You can choose different, more than one gene. Okay, so here, this is just showing me which, so this didn't change, okay, when I put my gene. And here, what it shows is where the gene is expressed. So this is mirroring this. And wherever you see spots, it means in these cells, the RNA sequencing result of these cells, the gene expression is very high, which means the gene is most expressed in OE1, almost exclusively. If it was expressing embryo, you should see a lot of blue dots here. You see very little. Okay, and then I think now you can examine the differentially expressed genes, if I'm not mistaken. So no, I don't really know how to use this. I hope there's like a tutorial where you can learn, but yeah, sorry about it. This is not a feature that's always used. It's quite new for me. Okay. So yeah, so this is the data you're most interested in. Then in day three, we see the gene is expressed in OE1, and in day five, let's see if this expression is still there. Yes, it's in again OE1, but now it's a bit in OE2, very little. So if the expression is very, very strong, then you see very big spots. Let's see how it is in day seven. So this is day seven. Okay, in day seven, expressions are changed here. So now the gene is not expressed in OE1 anymore, you see. The expression is decreased. So now it starts to express in embryo, but not every part of embryo, especially this part of embryo. So I'm wondering if this part of embryo is grouped with an already known genes expression. So I examine, let's say, level 3 now. So you see, in level 3, we see embryo in many levels. And here, for sure, OE1 expression overlaps with second cluster. And I'm wondering what second cluster is. So this is PDF1 expression. And this is protoderm of embryo. So PDF1 is already known to be expressed in the outer part of embryo. So then OE1 is probably also expressed in the outer part, not vascular part of the embryo, for example. It's so nice, right? I mean, embryo is extremely small, and you have a now idea where it's expressed. So what you can think, since this is responsible for synthesis of flavonols, for example, quercetin, then I can immediately hypothesize in the growing embryo, I should have uh, quercetin in the very outer part of the embryo, not inside. Why? Because the gene is mostly expressed in the outer part. Of course, this can change as the days change. Like in 9 days after pollination, maybe it changes. Okay, so this is how to use the thing. We can also show this data in different styles, like I think the violin plot is really nice, but it's a bit complicated. Let's do it level 2. So level 2 violin plot. You see in embryo causal endosperm OE1. So you see the expression of the gene mostly in the embryo in 7 days. But if you look at 3 days, for example, back again, the same data but with violin plot now. You see a very strong expression in OE1. Almost, you can say it's almost exclusively expressed in OE1 at day 3. Okay, thank you. See you. Bye bye.